Great. So I'll jump right in and do the first couple of slides and pass it over to my colleague, Dr. Jaden Lee. So Zach Brooks from UGenome. We make licensed software tools for genomic medicine. I'll talk a little bit more about the team here in a few slides. We'll go to the next slide at this point. Now, if for some reason you have to walk away, get a text from a family member or loved one, and you have to step out of the room, we always try to give a preview slide or a summary slide. So a couple of highlights. The founders have been at 18 startup companies with 18 million raised. In our first year of business, 2023, we were in revenue and we're ranked number 3 biotech startup in Arizona for 2024. We have 2 leading products right now and 2 services. You can see some of the team below. Jack Florio, you might recognize him. He's in San Diego, runs the new fund. We were born from University of Arizona Technologies, but a lot of our technologies are now developed in-house. We're part of NVIDIA, Founders Hub, and Activate. Next slide, please. So the 2 things we'll talk about today are 2 problems. One is gene editing analysis, and the solution we are calling it in-house, at least Genome CRISP. And the second is cancer variant detection. We're calling that Genome MAX, and this is intellectual property we licensed from University of Arizona. And these are the 2 applications we're working on right now. Next slide, please. So the problem is off-target analysis. And if we just dive into the problem for a second, ask yourself if you know anyone who hasn't been touched by cancer in some way. So the very specific cancers we care about are the ones driven by massive variants. So we're just highlighting 3 here: pancreatic cancer, lung cancer, colorectal cancer. From a descriptive analysis alone, you can see the percentages of people die once being diagnosed are extremely high. If we highlight 3 folks we probably recognize, it's Steve Jobs, Susan Wojcicki, I think this is her pronunciation. She passed away just a few weeks ago, and Chadwick Boseman. So really a serious thing once you get diagnosed with one of these cancers. The other problem in this space are rare diseases. So 10,000 known rare diseases estimated every year. 80% have a genetic cause. Most of those are detected in childhood. Most of these rare diseases lack approved treatments, and we certainly want to be part of the solution in that space. So I'll pass the slides over to Dr. Jaden Lee, our Chief Genomics Officer.
So hi everyone. So here the first application of MAX is for off-target effects. So when you are in the process of developing gene editing therapies such as CRISPR technology, you want to make sure that your gene editing is appropriate for a specific site that you want to cut, knock in, or knock out. Right now, current standard is using a standardized reference genome, which has been derived from a few volunteers. So what we are proposing is that by utilizing a personalized reference genome, you can improve the accuracy of gene editing and also improve the efficiency of off-target analysis by reducing the iterations. The main concern with the off-target effect is that many times when you are trying to edit genes, it may affect more than just the site that you want it to affect. So off-target analysis is really important to make sure that your gene editing therapy is doing what it's supposed to do. And not only that, you want to make sure that the edited gene was actually the intended mutation. Sometimes it's difficult to distinguish between naturally mutated genes versus actually gene edited. So with utilization of a personalized reference genome, you can actually help reduce that type of off-target analysis and reduce the iteration and reduce cost. So that's what the MAX and CRISP is capable of doing. It generates that personalized tumor-specific reference genome so that you can reduce the off-target effect. The other application is overall cancer variant detection, and this can be applied in the sort of tumor profiling and tumor RNA sequencing. Especially the RNA sequencing reads are difficult to align. So that's where we have a personalized reference genome that can actually improve the alignment of RNA sequencing reads. Currently, the RNA sequencing reads, it's only about 70% that you actually get the reads appropriately. With the personalized reference genome, you can significantly improve that. So that's what MAX is here for. It is to generate personalized reference genome. Currently, the MAX sits in front of the entire bioinformatic process to actually generate the personalized genome that could be offered to aligner software. And the key thing I wanted to bring your attention to is the graph B and C. And the B is the one without the MAX-assisted reference genome, versus C is the MAX's personalized reference genome. And as you can see there, you see improved alignment with graph C, especially in the presence of indel mutations, which can be a little bit trickier to correctly map it. And not only that, MAX actually utilizes both DNA and RNA information to generate a personalized reference genome, because sometimes DNA mutations do not always get transcribed to actually generate a RNA, corresponding RNA. So it allows it to sort of differentiate that allele expression. Here's a quick interface. We are trying to make this into a user-friendly API where the users can upload just the raw sequencing file, and the rest of the process will be automated. And the output will look something like this, where it will generate index file and FASTA file that could be fed into the aligner software. And also it will provide some visualizations to understand and then analyze the genomic information.
Great. Yeah, thanks, Jaden. So our business model is this. We have aligned services to support our product development. So we only talked about one of our products today, which is MAX, and the other application of that, CRISPix. We also have another line of product in the pharmacogenomic space. So it's services plus tools. Next slide, please. This is one of our services we did a few months ago for Boehringer Ingelheim. So we've proven ourselves that we can do some of these good services and learn from them to develop MAX better. Next slide, please. Our greatest resource is absolutely our team. So Zach Brooks is my fifth startup company. Jaden can do a quick intro.
Yeah, I have been a clinical pharmacist by training, but now I am functioning more as a product officer here, mainly overseeing the development of our software products.
Great. Next slide. I'll just say, generally speaking, we have a phenomenal team. We're really proud of all of them. We put everyone in a position to succeed, and we're getting lots of great benefits from everyone who's part of our team. So next slide, please. In terms of our competitor, this is very general in terms of the genomic medicine software and genetic testing. We're assessing the market really deeply now. We have a fantastic product manager who's going through this and giving us a sense of the best who are out there. I mean, I certainly think we don't want to compete on the price space. We want to be a competitive price, but not so low that we undercut ourselves. So we certainly want to be more in the middle and determine the features we're good at and then what the market actually needs. So we're busy working at that right now. Next slide, please. In terms of the regulatory environment, we're considering which is the best for us. Right now, it's the one in the middle, clinical decision support, but we're very aware of the regulatory environment. So we can build our products modularly, meaning if we need to shift in a year's time, we've already kind of been thinking about the regulatory environment ahead of time as opposed to a retrofit. Next slide, please. Kind of the same thing with the previous slide. We're thinking ahead, what are the reimbursement codes that are on the market right now, trying to follow this as closely as possible. So working with some consultants to try to get better at figuring out how we can engage ourselves with different parts of the market, insurance companies and de-risking types of mechanisms to get paid. Next slide, please. In terms of the market, top down, the big number there, I think, is a 16.5% growth until 2030. If we look at the bottom up segments, certainly PGX analytical tools, therapeutics, some other things, we think the SOM from our analysis, this is one analysis, we've done many of them, 67 million by the end of this decade. Next slide, please. In another analysis, the bottom right number is a much larger number. This adds up all the tools we're in the process of conceiving, developing, researching, etc. Next slide, please. In terms of our fundraise, it would be 1.5 million, 8% interest. If the interest rates were to change by the Fed, we probably would change these numbers as well. Use of funds is certainly going to be into R&D, phase 1, and then phase 2, more sales and marketing, operations, legal, those kinds of things. And next slide. Yeah, with that, we're finished. Thank you so much for your time. We're looking forward to getting your questions. We're really impressed with the judges and panel that you guys put together. So thanks so much for your time today.
Well, you've got time for one question. I will here. So one of the challenges I'm perceiving in the market is, are people making money in these domains yet? Even with the the best of intentions and the best teams, how's the revenue looking?
Well, so this is definitely in the preclinical research space. Where we're making money, we see more readily is with our pharmacogenomic services and tools, and that's where we're pushing a lot of our efforts to make some income earlier on. We are making some money for bioinformatics services, but we see this as a longer term play in terms of our revenue generating activities. But we want to present on what was relevant to today's presentation, so this is what's relevant to today's presentation.
Great, yeah, and you have a lot of great information here. I think again, calibrating to the audience is hard to do in these kind of environments, but you might simplify these a little bit. But it does look like very well crafted slides and a well thought out argument overall.
Great, thanks. That's great feedback, and we can certainly work on simplifying and making it better every day.